This is for the microbiology of milk. The idea behind this is we are going to try to calculate how many bacteria per milliliter are in a sample of milk. Now we have obtained a milk sample. Usually what you buy in the store has already been pasteurized or ultra-pasteurized, so it shouldn't have any pathogenic organisms in it. And we've purposely taken the milk sample that we've had for a little bit, and I'll just say we've left it out to try to increase our chances of getting something in it. This is also going to involve dilution, so we have to take the sample from the original milk sample. We're going to put it on a plate. We will then pour agar on, so we're doing pour plates, but each plate is going to be at a successive dilution. So we're going to start with the full strength. We have the pipette. We are going to pull a milliliter out of the milk sample. So we're going to pull a full milliliter out, and we are going to dispense it into this first plate. You can see over here. So 1 ml goes into plate number 1. That is going to be a 1-to-1 dilution. You've taken 1 ml, and it's a total of just 1 ml. Now what we are going to do for the second plate, it is going to be a 1-to-10 dilution. So we are only going to add 1/10 of a ml, which I'm trying to see in here. So this would be a 1-to-10 dilution. We are now also going to take 1 full ml of the milk. We are taking 1 ml, if we go up to the 0 there, and we are going to transfer it into this 99 ml water blank. So if we're putting 1 ml into 99 ml, so 1 ml into this is now going to make that 100 mils. So we have done 1 to 100 for this dilution. Because this pipette was in the original milk sample, we are now done with it. Let me dispose of it. Because it's full strength, if you will, you don't want to now go into this tube with that same pipette because you would mess up the dilution factor in it, because it was more concentrated. So this is now diluted 1-to-10. So what we are going to do is we'll take a new pipette, and we are going to transfer 1 ml from, from the beaker. So this was a 1-to-100 dilution. We're going to take a full ml from that, place it into the third plate, which makes it a 1-to-100 because we took 1 ml. If we take 1/10 of a ml, that's going to dilute it, another 1-to-10 dilution. So this is going to be a total. If you had 100 and you diluted by another factor of 10, that would be a 1-to-1000. We are now going to go from the first flask into the second flask to dilute it even further. We don't know with our sample where the concentration of bacteria is going to be. We have no idea what the concentration is, so we're trying to go through a whole range, and hopefully one of these will fit within where we can count the bacteria. Remember, you count between 30 and 300. So now we will dispose of this pipette. And we will now use our third pipette. So you had your original sample, a 1-to-100. We just did another 1 ml into 99 ml. So that's another 1-to-100 as compared to this flask. Compared to the original, it's 1-to-1000. So we are now going to take a milliliter out of here, transfer it to plate number 5. And then we will take 1/10 of a ml out, further diluting into the final plate. Now we have previously removed our agar from the hot water bath. We are now done with these. So these will be disposed of. These are agar that has been in a hot water bath for doing the pour plates. So as you've seen previously with pour plates, you don't want to do this when it's too hot, but you can't wait too long to let it solidify in the tubes themselves. So I'm just checking the temperature, feel of it. So the agar is ready for us to go ahead and add in. So each plate will get a tube of agar. We're going to swirl it around to mix it. And then let the plate set to the side to solidify. Once it has solidified, then as always, what we do is we will incubate it, invert it, and we're going to incubate at 37 degrees. With the milk, it's fairly easy to do your best at swirling it. If you did not previously mark your plates for which dilution it is, make sure you do it before you mix the plates up, because that's going to be critical afterwards. You have to look at the dilution factor in order to determine how many bacteria per milliliter were in the milk. Because that's what our whole goal for this is, why we're doing this, is to figure out what is the concentration, which would be recorded as bacteria, number of bacteria per milliliter in that milk sample. So we'll let those solidify and then incubate at 37 degrees. In this first plate, this was a 1-to-1 dilution, so we just put 1 ml in, mixed it, and as you can see, this is what you would call a lawn, or would call, certainly, too numerous to count because you can't even see individual colonies on here. This is a 1-to-10 dilution, so this is diluted 10 times as compared to the first plate. You can see that it's a little bit lighter in color, so you can tell that it has been diluted. Still complete growth on it. As we move down through the dilutions, you can see it gets lighter and lighter. It may be a little difficult to see. You still have a complete lawn of growth at the 1-to-100 dilution. There is some growth at the 1-to-10, but not really individual distinct colonies, so we really can't count them. And by the time we're down here at the last one, a 1-to-100,000 dilution, there is no really true growth in there. There's where the agar was starting to solidify, but sometimes this is just the way it happens, that you can't necessarily count distinct colonies. But this actually is really good to see it gets lighter in color. That's what you're supposed to see when you dilute something. So it does show visually the effects of diluting the milk sample out.